So can I have the panelists come up? The way we're going to do this is I want the panelists to go ahead and introduce themselves because they can introduce themselves much better than I can introduce them. And I'm going to ask each of them to tell us who they are, what institution they're at, and then just a little bit about what their personal kind of specific scientific passion or interest in the UK Biobank data set is, and then we'll get into the deeper discussion. So I'll hand it over to Mark to start.
Okay, so good afternoon. My name is Mark Effingham. I'm the Chief Operating Officer for UK Biobank. I work for Rory Collins, who is our Principal Investigator and Chief Exec. I'm responsible for delivery of the UK Biobank study. So there's been a number of mentions made of UK Biobank today, but perhaps it's worth just a thumbnail sketch of what UK Biobank is for those who aren't so familiar with it. So Biobank is a prospective cohort study. We recruited 500,000 participants from across the UK between 2006 and 2010. Each were aged between 40 and 69 and attended one of a number of Biobank assessment centres around England, Wales, Scotland, where they provided a lot of information about themselves through both touchscreen questionnaires plus a study interview. They provided a number of physical measures such as height, weight, grip strength, spirometry, and they provided a number of biological samples, only one of which was assayed at the time, which was a hematology measure. All of the other samples went into long-term storage for when assays became appropriate. It is a closed cohort, but not a static cohort. So we aren't seeking to recruit any more participants, but we do engage with participants in an ongoing nature. So we write to them every year with updates on the kind of research that is being undertaken using the resource. We offer web-based questionnaires to try and phenotype areas which we can't get access to through the electronic healthcare record linkage. And we invite them to take part in various enhancement projects. So, for example, we're currently inviting back 100,000 participants to undergo a 4 to 5 hour imaging visit where they undergo MRI imaging, ultrasound for carotid artery, and DEXA scan, and do a repeat of the baseline measures. We opened access to the resource in 2012. We have over 8,000 registered researchers all around the world. So it is a UK national kind of program, but it really has international visibility. We have over 1,000 applications. We don't distinguish between academia or industry. It's an open access resource to bona fide researchers who want to do research that is in the good of public health. So I joined Biobank 3 years ago after 20 years working in industry, predominantly for IBM, where at the time we had all these great analytics and AI and similar, but what we found difficult was getting access to really good quality data. So one of the attractions for Biobank for me was really coming to a resource where we have data not just in spades, shovels and pitfalls, and a whole lot more. So that's probably enough about kind of a Biobank thumbnail, and I'll hand over to Shep.
I'm Melissa Miller. I'm in the genetics group at Pfizer, so I work in early drug discovery. And sort of, I guess, thinking about what my passion is. When I first started my PhD in epidemiology, I got really immediately interested in thinking about things that would predict disease, thinking about things like genetics or biomarkers or other factors of health, which we now think of as precision medicine, but of course I didn't know the term at the time, and so my PhD research focused on genetic epidemiology, and it has just sort of continued onward from there. And so, of course, really kind of thinking about that, the UK Biobank is kind of a researcher's dream because we do have the huge breadth of phenotype data that Mark has talked about, and we can really use all of that to be able to look at what the predictors of health and outcomes are, and really be able to hopefully discover things that can help improve people's lives.
My name is David Sexton. I am the Director of Scientific Computing at Biogen. And I've been in genomics now for 25 years, which is kind of scary. I had previously actually worked with Jeff at Baylor, and I go back to the days of positional cloning, so pre-reference genome. So probably not a lot of people in the room have been in that space before. And what really drives me is the data itself and making data fair, and that means a couple of things. The first one is making it available to everyone who would like to have access to it, but then there's also the acronym FAIR, which is making data findable, accessible, interoperable, and reusable. And that piece about making data interoperable and the multiple types of modalities of data that people will be dealing with, especially in the UK Biobank, is what really drives me. And the way that I look at this, the data analyst is typically the person who is giving the information that decisions are made on. So anything that I can do to make sure that those people have the data in the way that they need it so that they can make a speedy decision is really what drives me.
And I'm George Asimenos. I'm CTO at DNAnexus. When we started DNAnexus back in 2009, as much of a cliche as it may sound, our mission statement was to unlock the potential of DNA. And I feel that's very relevant right now because my passion is actually for us to create technology that will help unlock the potential of the UK Biobank and to actually accelerate the scientific discoveries that our customers are going to do on this very data set. So I'm really excited to be here.
All right, thank you, everybody. And for my part, just to remind people who either forgot or don't remember, I'm from the Regeneron genetics side, and one of the reasons for our having this panel here now is a variety of pharma companies, including Pfizer and Biogen, have partnered with us at the RGC and the UK Biobank to sequence all 500,000 exomes and to make that data available. I believe the goal is to have it available by 2021. And so I want to kind of start in terms of the questions about kind of getting more into the science for Melissa and David working for these pharma companies who are really committed and interested in this data set. For you personally, what is uniquely valuable about the UK Biobank? Why is this the data set to partner in this kind of consortial pre-competitive way as opposed to other data sets?
I think Mark talked about this a little bit already, but especially compared to other data sets that are out there, the breadth of phenotype data is just amazing. When everything has been collected and released, this will be the largest imaging cohort, and there are multiple kinds of imaging. So we're talking about having really nice detailed MRI imaging of the brain, imaging of the liver, carotid imaging. It would be the largest biomarker, the largest set of biomarker data that will be available, and I think there's about 30 different biomarkers. There's self-reported data, there's also electronic health data, and one of the other really great things is that, as Mark said, it's a closed cohort, but it's not static, so just about once a year, there are updates to the data in terms of people's electronic health records, so we really are able to look at things like what your biomarker data was at baseline, or what your brain looked like at baseline, and be able to correlate that with your health outcomes. And I think that's very unprecedented already just in terms of the phenotype data. And if you think about the fact that we're now going to be able to link genetic data to that, we really will have the ability to discover new genes that are associated with diseases where we previously wouldn't have had that information.
For me personally, it's really around the volume of data in the sense that data at this size will give us broad statistical power to understand effects at very small sizes. And having all of these modalities of data and being able to integrate them, I think we have an opportunity to learn about the genome. My uneducated guess is we probably understand about 50 to 60% of genome science, and the opportunity to really extend that is exciting to me.
So one of the things that Melissa hit on is the presence of both electronic medical record data and self-reported. I want to do just a quick experiment. Raise your hand if you've done any kind of study that involves self-reporting, like 23andMe or UK Biobank, or one of those applications where you're telling somebody about your health for a study. All right. Now keep your hands up. Now. Lower your hand if you didn't lie on any of that information. If you told absolutely 100% of the truth. All right, you guys are way better than I am. But so that was sort of an intro to the question of, and really anybody in the panel jump in, what is the value of self-reported data in the context of people who have also health data? Is it complementary? Is it not as good? Is it better? Like how should people think about how to utilize self-report data versus healthcare data and maybe in an integration of the two? So dive in,
So
Mark.
I think in my mind it's very complementary, and you would like to use both. In terms of the reliance you can place on self-reported data, it depends on the kind of the phenotyping. As I mentioned earlier, in terms of how we use web-based questionnaires to get additional data from participants, we've run questionnaires around kind of working history, around diet, and particularly things like diet. You just can't get access to that kind of information from electronic healthcare records. But you do need to understand that when people are answering those kind of questions, then they perhaps may not answer as accurately in terms of what exercise they did yesterday or what they ate yesterday, for example.
Or how many drinks a week they
Or how many
drink.
drinks a week. But particularly things like if somebody self-reports diabetes, you've probably got a pretty good confidence that's correct. If they report hypertension, then it's kind of less so, because there's a certain kind of interpretation and common understanding of the phenotype which you're having to assume. But I think the linkage to electronic healthcare records is passive. It doesn't need participant engagement. And for us, it's a way of following participants' health outcomes over time in a way that is scalable.
From my perspective, I would say that, as a data scientist, you would probably want to have as clean data as you possibly could have, that it would be structured, that it would come through a physician who actually understood how to diagnose these things. But in reality, you're going to have to deal with the data that you can get your hands on. And in a clinical setting, you're never going to go through a questionnaire of the size that you can give people to run through at their leisure. So I would say that there are obviously statistical methods that you could put in place to deal with that self-reported data. So I would say they complement each other.
So what do we think is most important in terms of how to respect the contribution of the participants, right? People have given over an enormous amount of data, maybe more data than on any other study that I know of. They have this deep characterization, self-reported data, electronic health record data, genomic sample. Like how do we respect those contributions, and in particular, how to think about data security, and how to think about data security in a context of making this data fair, as David says, making it as widely available and interoperable as possible.
Well, I would say data access controls are very important, is what we've heard. We're supporting the delivery of the results from you to other pharmas, and that's a very large set of people that need to interact. And as I understand it, some people have access to a different tier of information, raw information versus derived information and whatnot. So it's very important to at least understand the structure of the data, who needs to be able to do what, and code it with technology, so that at least you don't have to have reliance on your legal terms that say who shall not do this and that. You can guide people that are using the data to make sure they're within the right program.
The Biobank participants, so Biobank is Biobank because of the altruistic nature of the participants that have contributed their time, both the baseline assessment. As mentioned, 100,000 have been invited back to spend 4.5 hours with us with no kind of feedback or similar. And I think when we hold participant engagement meetings, which we do on a monthly basis, one of the key things that participants want to understand is what is now known, given my contribution to Biobank, that wasn't known before. It's actually quite difficult for us to answer that kind of question. We've had 500 publications now released based on research on Biobank data. It's very difficult for Biobank. We don't do research ourselves. We're effectively providing a resource for the research community to use, and it can be difficult for us to interpret what constitutes good research or what constitutes bad. But providing that kind of feedback loop to participants is really kind of important. I think the data security, the approaches that we take in terms of how we provide data to researchers in a de-identified way, I think that's kind of taken for granted by participants that we do that, and we do that kind of well. but they really want to know how that data is being used and how it's supporting research into areas that otherwise wouldn't be possible.
So I will say one of the things that's most exciting to me about this data set is just the availability, the idea that, of course, people have to have a bona fide research application, but also, people can publish on the sort of aggregate non-individualized data that results from that. And that I've already seen that echoing a bit in the genotype and the imputed data deliveries. And I think this is going to become a data set that, junior scientists really all over the world are going to be able to use. So to me, that's like something that's just really exciting, and I think speaks to going back to those participants and saying, see, we are really treating this data well, and we're getting as much value as we can out of it. So that to me, that's very exciting. Let me ask you, the phenotypes are very rich, and one of the things that I learned in coming to the RGC and getting more into phenotype data is for all of the complexity of genotype data that we've heard a lot about today, right? I mean, how many different talks about variant callers, can you have in one meeting, right? It's still like a rich and interesting thing, but the phenotype data is much more complicated. So again, sort of open it up to anybody to dive in. How do you think about, or what are the challenges with trying to attack the problem of extracting from such a rich resource really computable phenotypes that you can do something with?
David and I were actually just talking about this, and I was talking to somebody at DNAnexus about it during the break too. I feel like at Pfizer, this is definitely still an ongoing. We're still trying to solve this in some way, which doesn't involve us manually searching through each field, which is kind of what we're doing at the moment. And I think one of the things that would be really useful going forward is to get all of the different groups that are working on ontology to sort of have, to sort of work together and to think about what does a good ontology for the UK Biobank data look like, and have that be something that can be returned to the UK Biobank. I think the other really interesting thing about the UK Biobank that maybe doesn't solve the ontology, but kind of solves working with the phenotypes and how people derive the phenotypes, is that when you publish data, any new variables that you've derived, you return back to the UK Biobank. So I think that's another really great opportunity to be able to say, I'd really like to derive this disease, but I don't know where to start. And so quite possibly somebody has already done that work and returned that data back. So that's a resource that we can all continue to use and not have to sort of reinvent the wheel each time. And there's already pretty good examples of tools where people have done that. Stanford has a biobank engine where they have made all of their derived results available. The Broad has done something very similar, so certainly those efforts are underway, but I think there is a lot more work to be done there, especially given how many just sheer raw volume of phenotypes that there are.
And I would echo the ontology piece. I think being able to go in and all of the data sets would conform to a single ontology, and you'd have a way to roll data up to the highest level of a disease or a tissue type that you want to be able to. I think those are very critical things to be able to do. Then we also have obviously image data that would be coming in as well. So there are complex phenotypes that could come out of a combination of just the questionnaires or the clinical information along with the image itself. So I would see people, providing a way for people to create those complex phenotypes and then also feed those back as they do their modeling back to the UKBB would be one of the more difficult things that we'll have to do.
I think phenotyping is one of the more kind of difficult areas, and it's certainly got our focus over the next 3 or 4 years in terms of how we approach better phenotyping of the resource and starting to adopt some of the kind of ontology approaches. So there's mention of kind of FAIR. That's something that's very important to us. I think as the resource has grown over time, when Biobank kind of completed baseline assessment, effectively all of the Biobank data could stick onto a USB key. Since then, we've done activity monitoring, we've done cardiac monitoring, we're doing kind of imaging, and we've now got a very rich and diverse resource. And it can often be quite difficult for researchers to know what actually information is in there that's going to support the research, and that's something, again, we're looking to try and improve. We've got a team that sits in Edinburgh underneath our chief scientist, Cathy Sudlow, who are spending a lot of time looking about kind of algorithmically derived outcomes. So how do we start to take the secondary care fields, primary care data, and what is the right combination of codes which would indicate diabetes or subclassification and similar. We've done that for stroke and MI, we've done that for diabetes, and we have a plan to extend that across other areas. It's not saying it's the right way of doing it, but it's saying it is a way that provides some level of standardization, and we're publishing those algorithms as kind of open publish, which researchers can take and work with themselves.
Oh, I was going to say, for those that haven't had the opportunity to work with UK Biobank, life will never be the same once you take a look at the data set, because when you're used to having a drop-down with only 5 different phenotypic attributes to choose from, imagine you have 3,500, and some of those have encoded values, so there are actually tens of thousands of things in disguise, and there's multiple measurements for certain things, or diarrhea is now spelled with an O. So as you approach this, I think from a technology standpoint, and try to solve some of these challenges by adding text search and lexical synonyms and whatnot, you realize that now you have multiple sources of information. There's interviews, there's touchscreen displays and questionnaires, there's health records, and they all point towards a certain direction for do you have a particular disease or not. So I actually think that UK Biobank may need a model of its own, as opposed to try and learn from some of these other models that don't necessarily capture the true richness of this information.
I think that's a key point. So if we think back to Biobank was kind of incepted in the early 2000s as an idea, this is kind of pre-iPhone, the kind of technology and approaches we had at the time is quite different to today. And a lot of the taxonomies that underpinned Biobank were effectively that they weren't open source kind of versions available at the time, so a lot of them being internally kind of driven. There's some work we're doing at the moment looking at the likes of kind of EFO and OMOP and similar to try and do a mapping of those onto a standardized ontology, which again, we're not looking to try and direct, but we're trying to provide a kind of a baseline that different research groups can utilize. I think this becomes important when we start looking at return of data, such as summary statistics. When someone's done a GWAS for a particular phenotype, how do we then compare that to somebody else's similar GWAS for the same phenotype? Is it the same thing or is it different?
So I'm going to give the audience a chance for questions, but while I want to give you guys a minute to think about what questions you might ask, I want each of you to answer specifically what is the one thing or the two things that you would like to see added to UK Biobank. We've heard about the incredible breadth of both the EHR data and the imaging and genotypes and exomes soon, and there's a whole Genome Vanguard project, and there's going to be an enormous amount of data, but what isn't yet envisioned for UK Biobank that you each individually would like to see? So I'll start with George and come towards me.
Well, I think wearables and Apple Watch and those kinds of things seem to be...
It has to be an Apple Watch. It can't be.
Seems to be, I think the most easily collected and untapped resource. So as the more, science experts work on the real EHR, I think there are sort of hidden information that you drag with you as you walk through the universe might be proven useful in surprising ways.
So wearables is actually included, so that is coming. So that's and really, multiple modalities of data. Looking at things beyond just whole exome or whole genome data. Looking at things that get at chromatin or get at methylation are critical, and really exploring the whole central dogma. You can certainly see regulatory information being extremely critical here, especially in common disease. So the more information that we get in depth about biology, the better we will be with this data set.
You guys might have covered almost everything I would have thought of. I guess the one thing in terms of multiple modalities, I know there's been some efforts already being made towards this, but to think about other omics data that we're not measuring. We've talked a little bit about broad scale proteomics data. Obviously, methylomics data is already being done, so I think that would be one particular thing I think that could be added on as well as I know that there were blood samples collected from anybody who did a follow-up visit. So to be able to have the repeat biomarker data that's already available measured on those individuals would be great, just to be able to look at how things change.
And actually, we've already done that. So that is part of the that will be part of the biomarker release, looking at that longitudinal change. I mean, so I mentioned before, Biobank doesn't do research ourselves, and our kind of approach is tell us what to build, and we'll build it. We are thinking about participant kind of generated data. So how do we harness the fact that a large proportion of our cohort is quite technology savvy and probably do have a number of kind of wearable devices, and can we start to kind of capture that data, for example, to record activity over prolonged periods of time rather than just for kind of 7 days. We're very interested in new other kind of assays that can complement all the work that's been done around kind of sequencing. We're actually holding a workshop later this month to look at proteomics, metabolomics, what's the current state of maturity. We're very interested in doing cohort-wide assays. So data, we have kind of a fairly low bar of entry in terms of access applications. Samples are depletable, and our intent is to try and turn samples into data that we can then make available to researchers across the board. So certainly doing more kind of omics is very much of interest.
So for my personal interest, I mean, there is an enormous amount of imaging, but this is very interesting to me and to, I think, everyone generally. But I would love to see an expansion of imaging and different modalities of imaging and imaging for different purposes. But obviously, that's a huge commitment from the participants. So, again, recognizing that, there are real people behind this who have to do some real work to make this data set.
Just to say on that point, we will be starting in January next year with repeat imaging, so inviting back participants. I would say our participants are very giving. The fact that we have people who travel down from the northern parts of Scotland to Newcastle, kind of a 5 or 6 hour journey to come and spend 4.5 hours in quite noisy machines is kind of a testament to the kind of altruism. And we're going to be inviting them back to come for a repeat assessment so we can start to see kind of changes over time.
Oh, that's fantastic. All right, so I want to open it up to you all. I mean, we have a master class in all things UK Biobank here. It is really, to my mind, the single most important genomics data set right now and probably for the next 10 years. So now's your chance to ask any question about the UK Biobank or about the sequencing efforts that relate to it. So bring it. Who's got a question?
Can you state your name and your organization?
Ralph Snyderman, Duke University. So this obviously is a fantastic, big, broad project, and as you said, it is really the standard right now. I just wonder, what do you think of the role in the interaction of other smaller projects, such as the Baseline project that Duke, Stanford, and Verily are going to be doing? 10,000 patients, all types of mobile devices, being followed more closely in 2 medical institutions. Do you see this as something complementary or superfluous, or, I know you won't say that, you're polite, how do you see the interaction between so many precision medicine initiatives? Virtually every country has one. How do we get them to build and synergize upon each other?
So I don't think they're superfluous. I think they are complementary. And I think from a UK Biobank perspective, we set out to do 500,000. We would have liked to do more. So the more people that we can phenotype and understand their health outcomes, the better. I think there is a balance around how we try and kind of harmonise some of these efforts, particularly, for example, from a biobank side, the work we're doing around sequencing, how can we make sure that any kind of research outputs can be brought together with other cohort studies, such as the work that Genomics England are doing, but also kind of international efforts. So we try and do that on a kind of proactive basis in terms of reaching out. There's probably much more we could be doing. But again, we're kind of focused in terms of delivering on Biobank's priorities whilst also trying to kind of play nicely with other approaches.
One of the things that we've noticed in pharma, and I'm sure academic researchers have noticed this as well, is that while I think the UK was probably one of the first countries to do this, they are certainly by no means now the only. So China has the Kadoorie Biobank, which I think in overall size is very similar, although we talked about at lunch, I don't think they'll have the electronic health records. I know Qatar is doing something very similar, and that's just a couple of the countries that I can think of offhand. And what we know already from genetics is that we find very different things. We find very different disease harboring variants, very different genes that cause diseases in different populations. And so I think sort of the more wide-scale efforts we have like this, be they something like a 10,000 person cohort with more phenotyping, or biobanks from other countries, I really think the more we're able to learn, and I think something that would be really great, you guys obviously really set up a good model. It works well. The questionnaire was administered well. The data is very good. That, I think hopefully as some of these other biobanks are getting off of the ground, hopefully they're reaching out to you guys in terms of how to operationalize this, because then potentially maybe we do have some data that we can, try to use together, both in terms of sequencing and in terms of phenotype collection.
And I would restate that harmonization and integration of this data is important. We heard about all of us as well today, so that's going to be a very large cohort of data. And what I would hope is that some of the governmental organizations that are backing these consortia would actually get consortia together and say, can we agree on some common standards and formats of data, even down to formats of data, so that it will make it easier to integrate these disparate data sets.
I was going to say, yes, every approach is distinct enough to have a reason of existence. If you think historically from HapMap to Decode, even within the UK, we have UK Biobank, Genomics England, Browning Bradford. It seems that everyone seems to be focusing on a slightly different aspect that increases the entropy of information and makes some contribution one way or another. I could envision that the baseline project creates an understanding of who is perhaps a healthy background so that you could then create better association studies within the UK Biobank. So you can take a learning from one data set and apply to the other one, or use one for validating a hypothesis that you generated in a more disease-focused, smaller cohort. So,
one of the things that I'm very interested in is ancestrally heterogeneous sets. So I think one thing that's very important is if the idea is, well, we've done this in UK Biobank, we've done it with 500,000 people, and maybe there's other efforts towards 5 million now. There was something announced this morning, like, oh, well, that it's sort of done. I think that gets it wrong, right? Like, you have to really think about what are the things you're going to discover within a population, and different populations harbor different alleles, they harbor different phenotype enrichments. This is very well characterized across the field, and so I think there's enormous value in having a lot of different studies, particularly if they can be heterogeneous in terms of ancestral diversity, as well as heterogeneous in some of the ways that they're collecting data. Other questions? Looks like Mark has a question.
Hi, I'm Mark Mooney from TagBio. This sounds like an amazing study, and I was really interested to hear about it. I'm also curious, so you have 500,000 people and you develop all of this work on them. Do you inform the population within the UK Biobank of the results, or do they take action in response to things that have been done in research? And how do you deal with that kind of communication to your population? And then, of course, if you communicate to them, do they change their activities and alter the way they respond to information as well? Because they're a special kind of population.
So the simple answer is we don't. So we have a very clear principle around no feedback. So when a participant originally came to an assessment centre, they walked out with a piece of paper for their efforts which had their height, weight and BMI, and that was it. We have a clear principle about no feedback. So if there is research that's done on genetic data that reveals particular heightened risk to certain diseases, we wouldn't feed that back to individuals. We do, as part of the imaging program, and put a lot of time and thought into what's the right ethically, scientifically sound way of dealing with potentially serious incidental findings. So where we do observe things whilst a participant is being imaged on an MRI scan, for example, we will review that. If we believe it to be potentially serious, we will write to both the participant and their GP to have it followed up. But that's a very specific thing to the imaging program. We don't feed back otherwise.
Other questions? We have one over here.
Hi, I'm Jeff Basine from Zymer Research Corporation. My question is about what issues are anticipated in terms of multiple testing and false discoveries, and perhaps in the broader philosophical sense, the usual way we do research has a hypothesis up front. So in this case, is this a resource where we can go in there and test a hypothesis, or is this actually more of a data mining approach where we just hope to find things? And then how do we address the concern of if we find things we weren't looking for, it may just be due to random error or false discovery?
Jeff, maybe you should say something.
So personally, I don't worry about the multiple testing issue because, first of all, this is a resource that a lot of different people are going to approach with a lot of different questions, right? If there's one thing that I think, the thing that's very exciting to me, and I think one of the reasons why this resource is having a lot of impact is kind of any bona fide research program can approach it. So if you have a data mining approach, you can make an application, you can do that. If you have a hypothesis-driven approach, you can make an application and you can do that. And then, given the previous discussion about having other data sets around, I worry less about multiple testing in a universe where there are a lot of different cohorts where one can do independent validation and meta-analysis. And so the idea that, well, maybe I was just rooting around and I pulled a bunch of noise out because humans are great at pattern recognition and I really wanted to see a signal for a drug. Then you go and look at one of these other studies that has similar data and you see if that replicates there, and if it does replicate there, then I'm quite confident in it. So I think it's a smorgasbord of research plans, and I think it's a little bit of everything.
I think when Biobank Access opened in 2012, it was probably geared towards the risk averse. So it's a fairly new resource, and maintaining that kind of contract with participants about how data would be used. So I think when it first opened, it was more focused on hypothesis kind of testing kind of research. We've kind of extended it and are very supportive of hypothesis generating research, but it needs to be within a prescribable scope of an application that can determine what is the benefit to public health. We do see a broad range of researchers using the resource, from kind of pure epidemiologists through to actually more technology-oriented organizations who are trying to kind of develop and optimize kind of machine learning techniques, for example, for image pattern recognition. So we do have a broad range of research that the resource is now supporting.
I think, in pharma companies, we're interested in understanding target identification and validation, so that is very hypothesis-driven in support of our research programs. So at least in our space, I would say that we're requiring data analysis plans internally that have to be written up, and someone has to approve those before they would really get access to the data set. And while there might be a mining application here, I think we would downgrade doing that, at least at the pharma level, where we'd actually be looking for something that supports our programs.
Any other questions? Anyone else? So I have one last question if there's no other questions. What's next? And I ask this very generally, and I'd be sort of curious to hear everybody answer. And it's not just what's next in terms of UK Biobank or what is the next project or what is the next sequencing thing. You all here are really on the front edge of what's going on in terms of this large scale genetics. So maybe we'll start with Mark and go the other direction. What do you think is next?
Well, I'm certainly intrigued by the announcement this morning by our Secretary of State for aspirations for a 5 million genome sequencing over the next 5 years. I think for us in Biobank, I think there's a significant focus on data linkage. So we have pretty good coverage at the moment. There's work underway to extend full coverage for primary care data, which we think is really important. I think the work on the outcome adjudication algorithms is really important. But then it is looking about how do we start to extend the data available within the resource into other kind of omic areas is very much of interest right now.
I think I maybe have already mentioned my what's next, and I think that is, and Jeff has as well, the sort of the fact that this now seems to be catching on as a worldwide phenomenon. So I think probably, hopefully, in the next 5 to 10 years when we have this conversation, we'll be talking about being able to look at the genetics from, hopefully 20 or 30 or more different biobanks around the world, which again will be differently enriched in different variants and different diseases.
More and better. I think that's the general answer for everyone. But really, I think biology is getting to a data problem. And I think physics had this problem maybe 10 years ago. You're a physicist, you can tell me. And they sat down and made the hard decisions and came to the point where they could actually organize their data. And I think this field is going to have to do the same thing. And I know there are varied interests that will want to have a say in that, but I think it makes sense for us to sit down and work this out now before the deluge actually hits us.
And for us, all of us is coming up. We're involved, and I'm excited to apply the same technology that seems to be applicable for large cohorts. But also, as you will hear in the next couple of talks, more disease-specific genome-phenome cohort generation. So that's also something very, very exciting for our point of view.
Yeah, and for my part, I would say, I generally agree with the panel. I think there's going to be more of these kinds of studies. I think this is actually the most valuable thing we can do, and being able to have other studies where you can do replication in places where, if David's vision is realized, where we can really harmonize this stuff together and build like bigger, more ancestrally diverse cohorts, that seems really important. So if there's maybe one last question in the audience, one quick one, we could maybe take it.
I'm on the healthcare delivery side, Duke Health System. If you had asked me that question as to what I would hope is next, what I would hope is that based on this information, we would start developing the predictive tools that were envisioned back in 2000 when all the excitement about personalized medicine began. So it is now 18 years. I am in the process of writing a review of what healthcare 2030 will look like. And one of the reasons I came here specifically is the hope that by 2030, this information would start giving us at least some clues as to how we could make healthcare better, enhance health, well-being, and mitigate disease.
Very well said, and I think we will close on that note. Thank you so much to the panel. Some people flew out pretty far to get here, so we thank you for that, and thank you so much for listening for so long.